Hello, this is Kevin, Craig, and Carly's Physiology project on nerve cells and anti-seizure drugs. Our objectives were describe the nerve action potential, including latent period, threshold, spike potential after depolarization, after hyperpolarization, and impulse propagation, as well as describe the basic physiologic mechanisms of common anti-seizure drugs as it relates to neuron membranes. Okay, here you see the basic anatomy of a neuron. Here is the body and the dendrites communicating with the other neurons. Here's the axon hillock where the action potential is going to travel down, and you can see the myelination of the sheath here, and in between the myelinated areas are the nodes of Ranvier, and it will progress on to the other synapses. Here is the physiology of a neuron, the actual action potential. You can see the voltage versus time. And here's our stimulus. Initially you're going to get depolarization, and then it spikes, repolarization, and the refractory period in which you can't initiate another action potential, and then it will return to the resting state.
Okay, impulse propagation of a myelinated nerve cell happens by what is termed saltatory conduction. As you may remember, the anatomy of a neuron is constructed so that the nerve impulse travels down the length of the axon in the most efficient way possible. Along the myelinated axon, there are sections that are void of myelin. These are called the nodes of Ranvier. As the action potential travels down the axon, it jumps from node to node, which allows it to transmit much faster than if it were to travel the full length of the axon. Next, we will explain the generation of an action potential. During the resting membrane potential of a cell, higher amounts of sodium are found in the extracellular fluid, and higher amounts of potassium and also protein are found in the intracellular fluid. This results in a relatively positive charge on the outside of the cell membrane and a relatively negative charge on the inside of the cell membrane. So now I'm going to talk about the steps of an action potential. An action potential typically starts with a resting membrane potential of the cell at negative 70 millivolts. It is stimulated by the amount of depolarization building to what is called the threshold potential, which is usually at negative 55 millivolts. This stimulus then causes complete depolarization of the cell membrane, which reverses its polarity and increases the membrane potential. During depolarization, opening of voltage-gated sodium channels make the inside of the cell more positive, and therefore the membrane potential increases to 0 and further overshoots to somewhere between 30 and 50 millivolts. This is referred to as the spike potential and is a value that is dependent on the strength of the stimulus. Repolarization then starts to return the cell's membrane potential back to its resting state. It is characterized by the closure of sodium-gated channels and the opening of potassium-gated channels, which decrease the membrane potential. In an attempt to do this, it then again overshoots its target, consequently dropping the membrane potential below negative 70 millivolts. This is known as hyperpolarization. Following this phase is the afterpolarization phase, which causes a refractory period. During this refractory period, backflow of the current action potential is prevented and generation of another action potential cannot occur. Shortly after this is the relative refractory period, in which the cell may produce another action potential. Soon after the refractory period, the cell returns to its resting membrane potential at negative 70 millivolts. Finally, the latent period is the period after a nerve impulse is applied to a nerve cell before the start of another action potential, and represents the time the impulse takes to travel along the axon from the site of stimulation.
Now I'm going to talk about anti-seizure medications used to control epilepsy mainly. So now I'm going to talk about voltage-gated sodium channel inhibiting drugs. Carly already went over how they work by blocking the sodium channel. The main drugs in this class are phenytoin, phosphenotoin, carbamazepine, oxcarbazepine, valproic acid, lamotrigine, topiramate, and zonisamide. And here are the drugs that affect GABA, which is an inhibitory neurotransmitter. So these drugs will actually potentiate the effect of GABA, thus preventing seizures. Phenobarbital, primidone, clonazepam, diazepam, and lorazepam. So here's more drugs that are GABA potentiating drugs. So these potentiate GABA by blocking the reuptake, which actually leaves more available GABA in the synapse to prevent the excitation and thus activation of the action potential and prevent seizures. So these drugs are gabapentin, pregabalin, and tiagabine. Okay, the final type of drugs we're going to talk about is the T-type calcium channel inhibitors. These drugs are going to reduce current through T-type calcium channels, which are pictured here in the cell membrane. The main drugs of this class are ethosuximide and valproate, which are both used for absence seizures, and valproate is used for just about all seizures. And that concludes our presentation on the physiology of nerve cells and anti-seizure drugs. Thanks for listening.